Good afternoon. Welcome to the fourth seminar of Hellenic Initiative Against Alzheimer's Disease. I, Ioannis Sotiropoulos from Demokritos, and Panos, Alexopoulos from University of Patras will welcome you. We will share this very interesting, I would say, debate between two great scientists all around the world. Panos?
Yeah, hello to everybody. I would like to make an introduction to the concept of bridges, which is an effort of the Hellenic Initiative Against Alzheimer's Disease to facilitate the dialogue between basic scientists and clinicians working in the field of Alzheimer's disease. The topic of today's seminar is blood-based biomarkers for Alzheimer's disease, scientific basis, clinical needs, and applications.
And indeed, we have with us two great scientists with a long and very well established experience on director of the Memory and Alzheimer's Treatment Center at the same university, the Johns Hopkins Bayview Medical Center. His research mainly focused on developing new treatments for Alzheimer's disease, especially psychiatric symptoms and relevant biomarkers, including sleep and circadian rhythm. And we're very, very happy to have him with us. And the second speaker?
Yeah, it's an honor that we have with us today Associate Professor Efthimios Dardiotis, who is the director of the Neurological Clinic at the University Hospital of Larissa, while he simultaneously serves as the scientific head of the local dementia daycare center in Larissa.
So thank you again for joining us, and the floor is yours. Temi, you are the first, correct? Thank you. You can start sharing your screen. Thank you.
So thank you very much. Thank you all for the kind introduction and the kind words. Actually, I am honored by participating in the HIAAD initiative. So the topic of my talk is the various clinical needs we have as clinicians dealing with patients with Alzheimer's disease, and how current and future biomarker may help us in our decision making. So this is an overview of my talk. First, I will refer to the need for having diagnostic biomarkers in order to help us with a definitive diagnosis of Alzheimer's disease, the early diagnosis, and also to estimate the conversion risk for preclinical stages of Alzheimer's disease or MCI, and also to help us in the differential diagnosis of Alzheimer's disease. Then I will refer to the prognostic biomarkers and also to biomarkers that may help us evaluating the staging of Alzheimer's disease pathology and use them as a follow-up measures. Also, we'll refer to the need for therapeutic biomarkers in order to assess the therapeutic response, either the efficacy or the toxicity of the new medications. And also there is need for having predictive biomarkers in order to estimate the disease risk in the normal population. Finally, we'll talk about some ethical issues raised by the use of biomarkers and also some other needs. So regarding the definitive diagnosis of Alzheimer's disease, we should say that Alzheimer's disease is a clinical pathological continuum with the first signs of amyloid deposits starting even 15 years before the clinical onset, and then they progress silently. And at some point, they trigger neurodegeneration with neuronal loss, axonal loss, and also loss of synapses. Clinically, we have a long period of asymptomatic stage of the disease. Then some symptoms appear. We have the subjective cognitive symptoms. Then we have behavioral symptoms, MCI, and finally, we have the clinical phenotype of the disease. So dealing with the definitive diagnosis of Alzheimer's disease, we should say that it is based on the sensitivity of the criteria we use. If we use pathological criteria as a gold standard, this definitive diagnosis comes very late. If we use clinical diagnosis, then the diagnosis is somewhere here. And if we use biomarkers, then the diagnosis is earlier. So we moved from the pathological diagnosis of the disease to the clinical diagnosis of the disease. Here are the criteria for MBI. And finally, to the biochemical diagnosis of Alzheimer's disease using the ATN system from the amyloid, tau pathology, and evidence of neurodegeneration. So currently, we use this type of biomarkers for amyloid deposits, the PET, amyloid PET scan, and also some measurements in the CSF, also the tau PET for tau pathology and some measurements in the CSF. And finally, for neurodegeneration, we have the FDG PET, some structural MRI, and some biomarkers in the CSF. So based on this ATN system, definitive diagnosis we have only if we have evidence of amyloid and tau pathology. And in this continuum, we have the clinical diagnosis somewhere here. So we move to this point when we can have earlier diagnosis of Alzheimer's disease based on the amyloid and tau biomarkers. And we hope that in the future, having additional biomarkers and also plasma biomarkers, there may be another shift towards the earlier stages in this AD continuum. Also, regarding predictive biomarkers, this may help us in predicting the conversion risk from the preclinical stages of the disease, for example, the subjective or when we have subjective cognitive symptoms, the MBI, MCI, to estimate the conversion risk for Alzheimer's disease. So challenges in these diagnostic biomarkers is the long prodromal stages of the disease, the variability in the clinical phenotypes. We have the visual variant of Alzheimer's disease, we have the language variant, behavioral variants, and also AD mimics and comorbidities. The previous mentioned biomarkers help us in the differential diagnosis between Alzheimer's disease and non-Alzheimer's disease dementia. So there is a need for other biomarkers that may help us in the differential diagnosis, for example, from other dementias, such as Lewy body dementias and other synucleinopathies using A-synuclein, FTD, vascular dementia, CJD, and dementias with mixed pathology, for example, DLB and also AD or AD and vascular dementia. Another category of biomarkers are the prognostic biomarkers, and these biomarkers that help us in staging the disease pathology at a specific time point. So in this continuum, again, it would be very useful if we had biomarkers that give us an image of the staging of the disease pathology and the severity of the disease at a specific time point. And then these biomarkers could be used in the follow-up. And another type of biomarkers are those that may help us in the prognosis, given that the levels at a specific time point may have a prognostic value for the short or long-term course of the disease. In fact, staging of Alzheimer's disease is nowadays feasible in vivo using PET imaging, amyloid PET, tau PET, and synaptic function PET. And we can see the differences in between the different types of the stages of the disease. However, we should admit that there is not a clear or strong correlation between the deposits of amyloid, for example, or tau deposit with the clinical severity of the disease. However, this staging of Alzheimer's disease is helpful to us in order to stratify our patients. The question is whether by using plasma biomarkers we can predict the amyloid PET and tau PET stages. And here is a recent study in a relatively large amount of participants, either cognitive unimpaired or participants with MCI or Alzheimer's disease. And the researchers used the combination of these biomarkers and found a significant correlation with the PET, amyloid PET and tau PET stages. However, there is still room for improvement to translate the group level results into individual level cutoffs in order to help us for clinical decision making. And it is possible that in the majority of our participants, this can be feasible using plasma biomarkers. However, some individuals may fall into this gray zone where additional biomarkers may be needed. For example, PET scan only in these individuals in order to categorize in which category they fall, either this one or this one. Another use of these severity biomarkers is to help us discriminate the prevalent pathology of Alzheimer's disease. And now that we are in the era of DMTs, we understand the importance of these biomarkers. For example, it is possible that patients with advanced tau pathology may not benefit from monoclonal antibodies targeting the alpha-beta deposits, and such treatment should be administered to individuals with low or intermediate tau pathology. We will see in the future if the information like this will be helpful to our decision making regarding which monoclonal antibody to use. Finally, we need prognostic biomarkers, having the levels of a specific biomarker at a time point in order to see the prognosis of the disease. And given the limitation of amyloid beta, which is not well correlated with the clinical severity, maybe synaptic proteins such as neurogranin are good candidates for having a prognostication. There are some good results in neurogranin, and we will see in the future if these results will be replicated in other trials. We also need biomarkers to assess the therapeutic response, either efficacy or toxicity of the medication we use. We know from the literature that many clinical trials have failed to reach statistical clinical significance because of the clinical endpoints. So it would be very useful and urgent to have a biomarker to be used as a surrogate of the clinical symptoms of the clinical endpoints in order to see efficacy of the medications that are tried. However, there must be a clear correlation between the levels of this biomarker and the clinical outcome, which was not the case with the amyloid deposit. We know that the clearance of amyloid deposit wasn't associated with clinical efficacy. Finally, we need predictive biomarkers to assess the disease risk in normal population. We know that Alzheimer's disease is a multifactorial disease with almost 40% attributed to modifiable risk factors and the rest attributed to genetic factors. So that the genetic landscape of Alzheimer's disease is complex due to large genetic studies with many common variants, less common variants, and rare variants, either protective or risk variants. So it would be very useful to have some kind of calculator to implement the genetic background of the individuals, and then to calculate the modifiable risk factors, for example, physical activity, diabetes, education, hearing loss, dementia, smoking, in order to have a score that gives us a risk of the disease in the normal population. With all this information, there are some ethical issues. For example, whether results from research studies should be disclosed to research participants. There has been much debate about that. It is possible that knowledge of the disease burden, amyloid burden or tau burden, and therefore the risk for Alzheimer's disease may cause discrimination, may cause stigma, psychological stress, may influence the access to the healthcare system, insurability, and also may affect the decisions for employment, driving, independent living. So generally, as I saw in the literature, disclosure of these results in research participants can be justified in participants with cognitive impairment, provided that these results are valid, are interpretable by clinicians, and are actionable in the sense that these individuals can participate in clinical trials or can have access to DMTs or can make the appropriate adjustment in their lifestyles. Finally, given all this complexity of biomarkers and also the increased risk of incident cases that are expected in the next year, we need human resources. We need doctors with an expertise in Alzheimer's disease and generally health professionals with an expertise in Alzheimer's disease in order to interpret all this information regarding biomarker use, to treat patients, to select the appropriate patient, to see and to detect possible side effects, and also to implement preventive strategies to the normal population. So we have a lot of needs as clinicians, and I hope Paul has some good news about how close we are to have biomarkers, plasma biomarkers, to meet all these needs. Thank you.
Thanks, Themis. You can stop sharing your screen. And we would like to invite Professor Rosenberg to share his screen and tell us, from his perspective, what is the great news on the discovery of blood-based biomarkers. You should unmute your microphone, Professor Rosenberg. I think we can hear you very well now.
Yep. And let me know, can you see my screen?
Yes, perfectly. Thank you very much.
Very good. Thank you for inviting me. And I'm embarrassed that I'm really the only person who is not Greek fluent on this call, but I'll do my best to struggle by in English. And I promise to not to take you on an Odyssey or the Iliad. So thanks for asking me to talk about one of my favorite topics. There's really been a revolution in blood-based biomarkers. It's not completed, but it's well worth paying attention to because I think it will be coming to a clinic near you in the near future. My disclosures, I do a lot of research and I do consulting to pharma mostly on study design. So in order to innovate Alzheimer's disease treatments, we need biomarkers. And Themis really pointed out a lot of the uses of this. To diagnose the disease early, for better differential diagnoses, to monitor treatment effect. And he reviewed the established biomarkers of amyloid and tau pathology. We've got CSF, you've actually got decreased amyloid and increased tau and phosphorylated tau, and PET, where we have a multitude of amyloid and now tau tracers. These biomarkers, however, are invasive and expensive. And in the past few years, I don't have a better word for it, there's been a revolution in the blood-based assays. I would say 6 or 7 years ago, they were close to worthless for the purposes we want. They're now pretty close to being very valuable. I'm no chemist. I'm imitating what my chemist friend told me. A couple of innovations in the plasma Alzheimer's assays has allowed us to measure extremely low concentrations. One is called the single molecule array technique, Simoa, developed by Quanterix in 2014. It's a digital immunoassay. It's similar to the ELISAs, the immunoassays we've done for many years, but they need relatively large reaction volumes, up to 100 microliters. And the problem is that everything's diluted and they can only detect sensitivity down to the picomolar range. Simoa allows them to get about three orders of magnitude better and get to be able to measure much lower concentrations. Each well has a single molecule. That's why they call it single molecule assay. Then at high sensitivity means you don't have to have as much labeling reagent. It means there's less noise. There's also what's called mass spectrometry. It's generally more accurate, but it is more expensive and labor intensive. And one of the assays that's already commercially available uses this. And just to put it loosely, IPMS includes immunoprecipitation and eluting with a peptide, and then mass spectrometry for the final analysis. It's really generally better than immunoassays, but it's a lot more labor intensive. Mass spectrometry is not a minor thing to do and not that easy to automate. And on the right, I show you a cartoon of a pretty standard immunoassay. And there's a single molecule. Things are so dilute, there's on average a single molecule in each well. So how do you apply these? So we know that a low ratio of amyloid beta 42/40 reflects high brain amyloid deposited in plaques. And we know this in CSF. We see similar reductions in plasma. Sorry for the redundancy. I mentioned the two kinds of assays. And for the mass spectrometry, the accuracy of the plasma assay was very high, 82 to 97%, whereas the Simoas was more like 60 or 70%. And so the mass spectrometry assay is now available in the US. I don't know its status in Greece. It's called Precivity. Very clever. In order to improve their detection, they include ApoE status, but there's no DNA. They do it through mass spectrometry, and they pick up the ApoE protein signal. So in the US, this is available. I haven't ordered it yet, but I plan to do so. It costs $1,250. When I put the slides together, that was 1,150 euros. Not cheap, but far less expensive than PET in the US. And so it's something to think about. It reflects amyloid. It doesn't necessarily reflect disease. Think of this as a way to approximate the amyloid burden in the brain, and so approximates PET or CSF findings for amyloid. So the caveat is that it's still not a huge signal. There's an 8 to 15% reduction of this ratio in plasma versus 40 to 60% in CSF. So it's just harder to detect, and there's more opportunity for noise. Both of these amyloid betas are produced by peripheral tissues, and so the brain signal is diluted. And so the plasma is a little bit of a less robust marker than CSF for biologic reasons. We're still working through how it varies in populations. So something that is even more remarkable, it has been phosphorylated tau. These new assays can detect it pretty well in plasma. It correlates well with CSF tau, with tau PET. It correlates with tangles on autopsy. So that first line is saying that plasma phosphorylated tau correlates with brain tau. So in a sense, it's an assay for tau and reflecting what's going on in the brain. But wow, it also reflects brain amyloid. This is where, to me, there's kind of a revolution. It correlates quite well with brain amyloid. And so the question is it a marker not just of amyloid and tau? Is it a marker of Alzheimer's mechanisms? Is it a biologic marker of the disease process? Which is a pretty profound idea. There are 4 sites that have been particularly studied where the tau is phosphorylated: 181, 217, 231, and 212. And there's a bit of an arms race right now, deciding which of these is the best of the bunch. Unlike the small differences in A beta 42 to 40, phosphorylated tau is increased a great deal in Alzheimer's disease as compared to controls. It's a much more robust signal. And in a number of studies, the diagnostic performance of p-tau217 is pretty comparable to CSF and tau PET. I will show you some graphs. So the gray area are the correlations between plasma p-tau217 and tangle densities in Alzheimer's. The gray is in Alzheimer's, and the orange is in non-Alzheimer's. I think it's actually in normals. And you can see it's a pretty good correlation between p-tau217 and tangle density with pathology in Alzheimer's disease, and correlates also well with concentration seen on autopsy. It's always hard to show receiver operating characteristic curves. It's always hard to make sense of them in a quick slide, but suffice it to say, what I'm showing you here is that it is excellent diagnostic performance between versus tau PET. There's tau PET versus amyloid PET, and versus tau PET. These are temporal regions on tau PET, and the top is with PET biomarkers, and the bottom is with CSF biomarkers. And it's p-tau217 is quite good at discriminating people who are amyloid positive from amyloid negative in MCI. This is really important because one of the most important things that with people with early memory problems is there's a strong negative predictive value with amyloid. If you don't have amyloid and you have MCI, you're probably not getting Alzheimer's disease. You might be getting another dementia, or you might be getting no dementia at all. So this is an important comparison. And as you can see, the upper bars are the mass spec assays. So you can see there's a really large difference, a 4- or 5-fold change of p-tau217 between the A beta positives and A beta negatives. That was using the WASH-U assay. And then there's a number of other assays, immunoassays, which are not quite as good. One from Lilly, I forgot which these other two are, but there was a comparison here of different assays. But the p-tau217 is a much larger signal than, for example, p-tau2-181. So the 217 is starting to look like a better biomarker for amyloid positivity. 231 may be the most sensitive earliest assay. People are just starting to work on this, but there are increasing signs that in preclinical Alzheimer's disease, this might be even better than p-tau217. And I think at this point, you've all been to enough Alzheimer's lectures to have seen cartoons like this, which are a model of how the biomarkers work. And suffice it to say, this with current modeling, p-tau231 seems to increase earliest and p-tau217 later. And just notice 181 is later, plaques are later, tau tangles are later than that. So there's kind of an order, a characteristic order of biomarkers changing. Moving on to neurodegeneration, neurofilament light. It's a neuroaxonal injury marker. It is sensitive, but not at all specific to Alzheimer's disease. It's elevated in most neurodegenerative diseases, including ALS, HIV, and atypical Parkinson's. It might be a surrogate outcome marker. The FDA has actually approved an ALS drug based on reduction of NFL in the CSF, an interesting biomarker. NFL, though, levels are strongly related to age and kidney function, and I'll get to that. And combinations of blood biomarkers may be better, and people have been moving toward a three assay combination: p-tau217, A beta 4240, and NFL. And then NFL is sort of a neurodegenerative control. So if you're positive on these two, looks like you're having an Alzheimer's process. If you're positive on NFL, it just says you have a neurodegenerative disease. And we are using this exact model in a grant proposal we are resubmitting shortly. Too much on this slide, and I apologize, but they're both A beta 42/40 ratio and p-tau are pretty good at distinguishing amyloid positive versus amyloid negative. And they're not wildly different. The AUCs are close to 0.9 for both of them in this recent paper published this year. And once again, I apologize, a lot on this table, but suffice it to say that p-tau 217 has excellent performance distinguishing Alzheimer's from non-amyloid dementias. And the likelihood ratios are really quite high, anywhere from 2 to 20, depending on which comparison you're making. But it actually right now, this is probably the single place where you can use most effectively. And once again, more recent validation of p-tau 217 versus amyloid PET. I apologize for a little redundancy, but this is a field where the results are absolutely rolling in every month. And I wanted you to see some recent replications. And you can predict future decline. This is the BioFinder cohort. It's a Scandinavian cohort. I forget if it's Swedish. And what you can see here, the PAC is a computerized cognitive test, just particularly optimized for detecting change in normals. And if you look at p-tau 217 quartiles, if you're in the lowest quartile, you're least likely to decline. And if you're in the highest quartile, you're much more likely to decline. And the same thing goes for mini mental status exam. So, and this is important because it may be useful for predicting prognosis in people who are cognitively normal. And here's the same thing, looking at survival to MCI or other cognitive impairment. You can also use it to monitor target engagement in trials. Here's the Clarity trial with lecanemab. And what they found was that on placebo, over 18 months, CSF p-tau increased. And on lecanemab, CSF p-tau decreased. But the same thing for plasma. The curves are the same shape. The signal's not as strong. But I think you're going to be finding this as evidence of target engagement in future trials. There are really important caveats, and this is why you can't just click on your computer screen and order your p-tau 217 today, but maybe tomorrow, or realistically, I think in a couple of years. The missing link is we don't have firm cutoffs for normal. We are lacking large systematic epidemiologic studies with diverse cohorts across broad geography. And I think it's going to be up to you and Greek authorities, really. Will you need a Greek study? How generalizable are these? But we already know that the levels are skewed upward by decreased renal function. Our friend Michelle Milkey, who is now Dean of Public Health at Wake Forest, but used to have the office next to mine, she published a very interesting paper looking at clinical confounds that confound p-tau 217 levels. And the highest number that she found was in a patient who had no Alzheimer's, but had decreased renal function with the creatinine of about 4. So they were in near kidney failure. So we're going to need to know all of these correction factors. We're going to need to know normals for different race and ethnic groups. And there's always a question with these of interlab reliability. Working on it and getting there, but not quite there. So conclusions. I can only say these are astonishingly good results. Believe me, I didn't leave out any negative ones. I just couldn't find any recent negative results. Plasma A beta 42/40 reflects brain amyloid, but plasma p-tau reflects both brain amyloid and tau. It's a marker of AD mechanism. The validation versus amyloid PET is very good, and these are already being used to screen participants for eligibility in trials. They're not quite ready for prime time, as we say in English now, but they're not far from it. My prediction is 2 to 5 years, but that is merely a guesstimate. There's my only Greek word, Ευχαριστώ. Thank you. And I particularly want to thank Kostas Themis, Abi Mogekar, who's our biomarkers expert here and is the one doing the assays, Barry Greenberg, who's a real thought leader in new treatment development, and Marilyn Albert, who runs our ADRC. I thank you very much for your interest. And Panos, should I stop sharing my screen now?
Yep. Thanks. That was really 2 great talks, a lot of information and a lot of clear messages from both of you on what we have in our hands, what we need, where we want to go. And I think the floor is open for questions. I see Kostas turn on his camera. So I assume we would like to have some questions from him. If not, Costa?
Yanni, thank you. I always have questions, but I'm particularly excited after 2 wonderful talks, setting the framework and bringing us up to speed. So I don't think my questions are necessary to help clarify. I mostly have a question for our colleagues in Greece. So let's say Paul is correct, that in 2 years, the data will be there such that these plasma biomarkers, take your pick, 217 we all think will show up first. I should add parenthetically that we actually have determined the cutoff in Abe Mogekar's lab here in terms of what level of 217 would almost definitively correlate with amyloid. But let's say we know it's 217, and let's say it's 2025. Who's going to do the determinations in Greece? And what do we as HIAAD need to do to set things up so when the data and knowledge is there, the country's ready to utilize this biomarker?
Themis, do you want to provide your view on that? And I'm sure others can jump in.
As far as I know, some labs here in Greece already use similar technique. We'll see in the future if they will implement this biomarker in order to have some first signals about these biomarkers, and maybe in the future using patients from different stages of the disease, we can have an imaging about the levels of this biomarker in patients with different stages of the disease. However, this technique is already known by Greek labs. For example, we sent neurofilaments in some labs in Athens in order to have an estimation of the neurodegenerations in some neurological diseases, for example, multiple sclerosis or ALS. I suppose that the same technique will be used in order to have biomarkers that reflect amyloid pathology or Alzheimer's disease pathology.
So just a quick follow-up. It seems to me we need to be proactive. Maybe this is a project for HIAAD to use some of the stored HIAAD samples so that 217 can be determined in plasma and correlations can be made with the CSF values of the more traditional biomarkers. Mm. I would strongly encourage you to talk to those labs and maybe think of working with Nikos, Karmeas, and others and say, in 2 years, we want to be ready. We don't want to be waiting for someone else to do this work.
Yes, this is very interesting. And we have this material. We have this. We have also CSF and serum samples from some individuals. We can store them and then have them determined to see the levels of the specific biomarkers.
Can I add a question? Because you said that the phospho-2 18 tau it seems to have a very good biomarker, blood, plasma, or serum. Do you think that it should be the same biomarker for when we are in the AD phase, or we should use different biomarkers in mild cognitive impairment, or when we have other comorbidities inside? So it seems from both presentations that we don't have a clear answer on that. So I would like to have Professor Rosenberg and Themis, also Kostas and everybody who wants to answer on that. So do we have different or better biomarkers for different stages of the disease, including MCI or BMI or BCI? Sorry. What is your feeling on that?
Right now, 217, p-tau 217 looks pretty good for many of the questions we're asking. And it looks better than p-tau 181. And the questions we're asking include differential diagnosis versus, say, FTD and non-Alzheimer's dementias. It's not bad for prognosis, and it's certainly not bad for approximating amyloid PET. So the competing biomarkers are the A-beta 42 to 40. It's hard to distinguish right now from p-tau 217. I'm not sure that it's better. And I think that what happens is if you use both, you get more bang for your buck. The one that's emerging for amyloid in normals is p-tau 231. And that's, of course, a very interesting area. It's something we're doing research in, but certainly clinically, we don't measure amyloid in normals because what are we going to do with the results? It's got far more profound ethical issues than measuring ApoE. Okay, and ApoE has enough ethical issues. But right now, if we found amyloid in people who are cognitively normal, what do? We don't do anything. We worry them. So I think p-tau 231 is in that space. But I think we're quite a distance from wanting to do mass screening of 70-year-olds. And that's really the ethical question is should we be doing that? It's almost as bad as doing PSAs, which we do.
Okay. What another question, and then if there are other questions, I can really stop. But when you are speaking about blood biomarkers, are you considering the free tau in the blood, or you want to go to the exosome, let's say, tau? How you see? We know the great work that Dimitris Kapogiannis and many others are doing in the States. We've been working with him, and we want to do more. But what is your feeling? You feel, because Dimitris sometimes say, look, I feel that probably blood exosome-free, let's say, markers would be better for diagnostic purposes, where if we go for brain pathology levels or status, probably exosomes will tell us more. So what is your view? And then I really would like to hear also Kostas, Panos, or Themis.
My view is that exosomes will allow us a much broader range of moieties we can assay. I am not sure that we need that innovation for amyloid and phosphotau. Maybe yes, maybe they'll be better, maybe no. But as you can see with the plasma markers, each one is a separate project. And we now have like 3 that might be useful. Whereas in exosomes, there could be dozens of moieties. Now, Kostas knows better than I how that research is proceeding, but I would just say, with exosomes, it's more like the sky's the limit for future discovery. With plasma, not so much. And in fact, there's some areas where plasma is really not working out. For instance, in Parkinson's, you really can't get a great plasma signal so far for alpha-synuclein, which is something we'd really like to get, just for example.
Yeah, this is a very deep issue, Yanni. I think the evidence right now regarding what are referred to as neuronal origin exosomes, including a recent analysis that we've completed from some of our samples where we compared plasma versus neuronal origin exosome content of the same moiety. I think right now the 217 is looking very accurate. So it's not clear that a more complicated process relating to exosomes is going to add very much, certainly when we're in the symptomatic phases. That's with the exosomes, or we should really be saying EVs. that are isolated to be of, quote, neuronal origin. I think the major advance, and I think our own vasomahierarchy with Ken Whitwer are going to be key to that, will be to improve the isolation of relevant EVs. So right now, L1-CAM and a few other neuronal surface proteins are used to pull the exosomes out and then characterize their content. I think using stem cells to be much more precise about what the right surface markers are for a target exosome is really going to be where the advance comes. And that'll probably speak more to contents of these much more precise EVs that relate to neurodegeneration as opposed to amyloid and tau. So I think exosomes are not there right now. I think they could be, and I think they will likely be there for more neuronal content related things. So tau, for example, synaptic proteins, other aspects of neurodegeneration, less so than for extracellular stuff like amyloid, which it's not entirely clear it lives in the EVs as opposed to sticks on the surface.
Vaso, any comment? I know you switch your camera on.
I'm listening. Thank you, first of all, everyone, for the nice comments. And I just want to add to Paul and Kostas what they said, that yes, it's a huge effort now in the field of EVs to actually find the specific brain cell specific markers in the EVs so that we will be able to isolate EVs coming from different brain cell types. Because what we think is that, of course, we can use them as a liquid biopsy instead of going to the brain, that we cannot take EVs from the blood. And this is not so easy, but we are trying. So it would be ideal. Like as Paul said, you have unlimited possibilities to see what there is either in the surface of the EVs or in the cargo of the EVs, that you can use them as biomarkers. So that's what we are trying to do. Hopefully, we will have some data soon. As I said, it's not easy. It's not easy because also the amount of neuronal derived EVs, which means they are from the brain in the periphery, it's very low. So we need to have
exactly, yeah.
Yes, it's very low. So we need to have a very good and very specific markers to be able to say that these EVs are coming from the brain.
And so here is where the IPSCs could.
Yeah, but let me give one more example where EVs may just an example of where EVs might be enlightening. We all know there's a lot of neuroinflammation in Alzheimer's and in most neurodegenerative diseases. And since I'm waving my hand, it obviously means there's something we don't know. Our plasma markers of inflammation really aren't very good. You know, IL-6, IL-10, and so forth. They approximate some of what's going on in the brain, but there's too much noise to use them as evidence of target engagement or whatever. With EVs, you might focus on microglia. You might say, let's look for EVs that are microglia derived, and it could help you look at mechanisms, hypotheses, and target engagement. And that's something completely inaccessible right now for anti-inflammatory drug discovery. Just to give one example.
Panos, thank you. Panos?
Yes, many thanks to both speakers for their very interesting and excellent talks. I would like actually to go back to the ethical questions. It is clear that blood biomarkers are less invasive and they are less expensive, and they will be surely useful in the differential diagnosis of people with dementia. My question is regards their role, the role of blood-based biomarkers in individuals with mild cognitive impairment, particularly in settings with no access to disease-modifying drugs. How we as clinicians will the clinicians benefit from such biomarkers in 2 to 5 years without having access to disease-modifying drugs?
Sammy or Paul?
So let me start. I'm sorry, I'm talking first, but let me start with just a quick reaction to that. So the essence of doing these tests and disclosing them is the pre-test education. And I really think that right now, it's not the doctor prescribing, it's the doctor and the patient and family deciding whether they want that answer. You know, we've done this for not me personally, but in prevention studies, we have done disclosures of amyloid status for a few thousand cognitively normal older adults. That's in the A4 study. It's a few thousand of them. 3/4 were negative and 1/4 were positive. And the overall disclosure process went pretty well. It wasn't that upsetting. Nobody suicided. I mean that seriously. There weren't a lot of adverse outcomes, and people welcomed it. But there was a research study, and they volunteered, and they consented, and they read 20 pages of crap before they could get their amyloid done. They knew exactly what they were getting into, and if they were positive, they knew that they might be offered a treatment. And so, my first reaction is, I don't think I'm going to do it without a treatment. But if a patient and family really wants it, I'll do it. But I don't think I'm going to be encouraging people to do it. And I think a lot's going to depend on how the treatment landscape evolves. Will these antibodies be felt to be effective? And will subcutaneous version administration work? So that's my answer.
Let me point out, Paul, you may or may not have seen this, but this morning, CMS, who is basically decides what Medicare is going to pay for, announced that if these drugs are, and we're really mostly talking lecanemab and maybe donanemab, if they receive regular FDA approval over the next few months, which is a strong possibility, that they will only cover them in the context of a registry. And it's still vague as to what that means, but companies will set up a registry, and that will allow CMS to create requirements. And it's entirely possible that they add biomarkers into that. So mild dementia, MCI, plus maybe a positive biomarker. And then a teen, they're saying, I think that means probably a specialized setting where the patients are determined to be eligible and where they are followed, MRIs, et cetera. So I think even with these drugs coming more openly on US market by the end of this year, there will be a lot of tight management of eligibility, of follow-up, and so forth. And the details remain to be seen. I think we'll probably have an opportunity there to include blood biomarkers, at least initially, so that, Panos, that might address some of your concerns.
Mm-hmm. Do you think, last question, I think, do you think that, based on what Kostas just said, do you think that will also open the door for an opportunity to invest more on even pre-symptomatic diagnosis, or let's say early, before even any symptoms? Because based on what you present us today, both Themis and Paul gave us a very clear things, more focus on diagnosis or early diagnosis. But what about pre-symptomatic phases? And we know that prediction is part of all health system would like to have in order to avoid, different risk factors. And that's something that at least precision medicine is supported a lot. So what is your view on that?
Yeah, I think we will need the same biomarkers or perhaps adaptations like p-tau231 there. Here's what I think is the most concerning finding of the donanemab study, and that's still based on seeing this in press release. They predetermined a secondary analysis of efficacy based on how advanced the tauopathy was. And the efficacy was greatest when the tauopathy was more advanced. It was really not efficacious when the tauopathy was minimally advanced. That, for me, creates worry that in a pre-symptomatic situation where there's only amyloid positivity and no tauopathy, that these drugs might not work.
Right. And in fact, I mean, we're part of a prevention trial with lecanemab, and the real question in that is going to be what's the progression of tau over time. With the donanemab findings, they're kind of fine-grained. They didn't include people, they didn't see a signal in people who had low tau, and they didn't see a very strong signal in people who had the highest tau. It was intermediate tau. And to be honest, you're going to need to translate this into blood-based biomarkers to make it work in practice. We're not going to be doing amyloid PET and tau PET on everyone.
That's true. That's true. That's very expensive and very invasive, and not health system can support it.
Yeah.
Okay.
So donanemab may have a tau range sweet spot.
Okay, that's what we'll probably will wait in the near future to have this criteria that where we can use these antibodies. Okay, if we have no other questions, once more we would like, on behalf of the Hellenic Initiative Against Alzheimer's Disease and also the team, the 5 people who are organizing this seminar called Bridges, we'd like to thank again Professor Efthimios Dardiotis and Paul Rosenberg for their 2 really great presentations and great discussion, and all of you for being with us. Our next seminar will be in 1 month, first Thursday, as always, of the month, and having another very more exciting question that can bridge basic translational and clinical practice. Thank you very much for all your attention and your participation. Thanks again, Professor Rosenberg and Professor Dardiotis. Have a nice afternoon.
Bye.
Goodbye.
Goodbye.